them into 1 molecule of glucose and 6 molecules of oxygen. Those are coming from the water, like that, in this water splitting, because of this water splitting reaction. And for a long time, they weren't certain of that fact. They knew the basic photosynthesis reaction, but some people thought that the oxygens were stripped off the carbons of the carbon dioxide. And what they eventually did is some very elegant tests where they used radioactive oxygen atoms, but only in the carbon dioxide, to see if you end up with radioactive oxygens in the water, in the oxygen gas, and you never did. But when they used radioactive oxygen atoms in the water, then those oxygen atoms were the ones that ended up in the oxygen gas. There it is, from the light-dependent reaction, water splitting is where you get the oxygens from. Go back. There we go. Now you don't normally see the oxygens bubbling out of the plant leaves, but because they just join the gas in the air and that's invisible. But some plants grow underwater, and then you can see the oxygen bubbling off as little bubbles. And this is a very common laboratory demonstration. They get an underwater plant like this and give it a lot of light, and the more light you give it, the more oxygen you can see bubbling out of the plant. All right, so where were we? So that was the source of the water splitting, is the source of the electrons to replace the one that photosystem 2 is giving into the electron transport system. And here I added that water splitting enzyme at the bottom of photosystem 2. So let's watch the whole shebang so far. Electrons are excited in photosystem 2, energized. Plastoquinone picks them up, shuttles them to the cytochrome complex. Water is split to replace those 2 electrons. The energized electrons are delivered to the cytochrome complex, and it uses their energy to pump up hydrogen ions, which ultimately are going to be used now to make ATP through the ATP synthase molecule. Now, notice the 2 energized electrons here. When they go to the cytochrome complex right there, I just showed them kind of disappearing, and their energy is being used. Their energy is certainly being used up to pump the hydrogen ion, but the electrons themselves can't just disappear. No molecule can just keep taking on electrons and electrons and electrons. So where do those electrons actually go? Well, eventually those de-energized electrons are going to be passed onward into more parts of the electron transport system. There are some more electron carriers here that carry the de-energized electrons away from the cytochrome complex. So let's drag them into the picture. Let's see. There go the energized electrons into the cytochrome complex. And so now those electrons have lost their energy. They're low energy again. What happens? Well, there's a second photosystem called photosystem 1, which is going to be involved in this process. And so it's eventually going to receive those 2 low energy electrons, but we need a shuttle, another part of the electron transport system, to ferry them from the cytochrome complex to photosystem 1. And this electron carrier that does that is called plastocyanin, right here. So unfortunately its name is similar to the other one. Plastoquinone shuttles between photosystem 2 and the cytochrome complex. Plastocyanin shuttles electrons between the cytochrome complex and photosystem 1. But they're all part of the electron transport system. Anyway, plastocyanin goes over there, picks up the pair of electrons, shuttles it to photosystem 1. And what's going to happen to them there? Well, photosystems are all about energizing electrons, and so that's what's going to happen. The sunlight It's going to also be shining on photosystem 1. So those pair of electrons are going to be re-energized. So they become high energy electrons again. And just like we saw before, they get trapped in the reaction center in the special pair there. And we're almost finished with the electron transport system. This, in the high energy electrons in photosystem 1 are going to be passed on to another protein located right here that's still part of the thylakoid membrane, but it sticks out into the stroma a little bit. And, well, I'll show you what happens. So the electrons move from photosystem 1 to this protein there. And that protein can bind to a molecule called NADP+, which floats around in the stroma. And with what's going to happen here is that the NADP+, floating around in the stroma, is going to take on that pair of energized electrons, and also in the process it's going to take on a hydrogen ion floating around in the stroma and become this new molecule NADPH, which is like NADP+, but now it's carrying a hydrogen. And that was the other thing that we were looking to create right here. That NADPH is in a way like ATP. It's a high energy molecule, and it gets its energy because it's carrying two high energy electrons. And this is also going to be used as one of the power sources for the enzymes in the Calvin cycle. All right, so let's watch it here. NADP+ docks on that protein. It picks up the two high energy electrons. It picks up a hydrogen ion floating around in the stroma. So now it becomes NADPH, like that, and it's released to drift over to the Calvin cycle. All right, so I think this is going to be the whole kit and caboodle. Let's look at it one time. Electrons are energized in photosystem 2. A water is split to replace them. The energized electrons are used to pump a hydrogen ion into the interior of the thylakoid, which ends up making ATP through ATP synthase. The de-energized electrons are shuttled over to photosystem 1, where they get re-energized by sunlight. Then they're passed on to this protein right here, which docks with an NADP+ and a hydrogen ion from the stroma, giving us NADPH. And so we've completed our task. We've changed sunlight into ATP and NADPH. How's that for mind-boggling? You may never look at a plant the same. Next time you see a weed, you're going, wow, you're pretty cool. All right, let me give you a couple of review questions to see if you've got it. So one of the key parts of this is this hydrogen ion concentration gradient inside the thylakoid. And that's important because that's going to allow the ATPs to be made when those hydrogen ions exit through this ATP synthase enzyme. What things contribute hydrogen ion to the hydrogen ion gradient? Like, who can tell me something in this scheme that helps make hydrogen ions there?
The enzyme that splits the water.
Thank you. Yeah, that's one that people forget. So I'm glad you mentioned it. I thought you were going to say cytochrome complex, and certainly that plays a role. That stuffs hydrogen ions in here. But what sometimes people forget is the water splitting is also important. Every time the water is split, you get two more hydrogen ions in the interior right here. So it's probably no accident that the water splitting enzyme is here towards the inside of the thylakoid membrane so that the hydrogen ions could get dumped in the inside and contribute to the gradient. If the water splitter was out here in the stroma, you couldn't use those hydrogen ions for the gradient. All right, so two things are actually moving hydrogen ions into the stroma, the water splitter and the cytochrome complex. But think about this, you also get a nice gradient if you remove hydrogen ions from the stroma. That also contributes to the gradient. So what is removing hydrogen ions from the stroma? I'll give you a hint. you would again point at the cytochrome complex because as it's putting hydrogen ions into the thylakoid, it has to be also removing them from the stroma, so that contributes to the gradient. What else is using up hydrogen ions in the stroma? Right, look over here. When those electrons flow to this protein, it's going to pull in a hydrogen ion from the stroma right there and attach it to this molecule. So you're decreasing the hydrogen ion concentration in the stroma, which again is good because that gives you a better concentration gradient to work with. You get more efficient ATP production. Sound good?
So the inside of the thylakoid also comes from?
Sorry, what's that again?
The inside of the thylakoid
Yes.
is also comes from, and the outside comes from?
No, the stroma is the liquid that's outside the thylakoid membrane. Did I call this the stroma by mistake? Maybe, perhaps I did. No, the stroma is the liquidy water stuff that's outside the thylakoid. So this is not considered part of the stroma. Let's see, any other kind of review questions I could give you here? No, not that comes to the top of my head right now. Should I go on?
So, overview, the photosystem consists of two pieces, and that's only within the thylakoid that the photosystem.
No, I'd say that there are two separate photosystems. There's a photosystem 2 and a photosystem 1. And there's also what's called the electron transport system, which includes plastocyanin, cytochrome complex, plastoquinone, cytochrome complex, plastocyanin, photosystem 1, and this enzyme right here. All right. And just incidentally, they've done experiments where they stuck little pH meters inside the interior of the thylakoid and also here into the stroma. And when you shine light on the chloroplast, you definitely see a much lower pH here than here in the stroma. And I forget what the exact numbers were, but it was something like pH 5 or 4 inside here and about pH 9 outside there, but only when light is shining on it, because if you think about it, light is what drives